Welcome to The Inquiry on the BBC World Service. One pressing question, four expert witnesses, challenging answers. In 1957, this was the sound of global superpowers locked in a cutting-edge scientific contest. When the tiny satellite Sputnik beamed its signal back to Earth, the Soviet Union claimed an early lead over the United States in the race for space. Today, there's another global scientific race, which some are calling Sputnik 2.0. And it could have an even greater impact on our lives. The new frontier is genetic editing. And this time, it's Chinese researchers who are claiming the headlines. They've been first to publish studies using a revolutionary new technique to edit genes in human adults and, just this year, viable human embryos. Scientists in the US and elsewhere are scrambling to catch up, chasing the prize of potential cures for diseases from cancer to haemophilia. And it's not just humans. Every living thing can be genetically edited, leading some to fear this race is a fast track to a dystopian future of rogue clinics, mutant species, and designer babies. I'm James Fletcher, and this week on The Inquiry, we're asking, is gene editing out of control?
Part 1. The Language of Life.
This is probably one of the most important developments in biology that I've been witness to.
Carl Zimmer has been a science journalist for more than 25 years, so that's a pretty big call.
Now you have this system for programming life.
He writes for the New York Times. Before we can ask if gene editing is out of control, we need to understand what it is. So let's start with the basics. The DNA that's in all our cells. It's a string of about 6 billion letters, which represent different chemicals.
I just think of DNA as being a Word document on a computer.
So if DNA is a huge Word document, think of genes as short chapters within it. Genes contain instructions for all our cells to make the molecules they need to survive.
So genes influence all of our traits. Each gene may only be responsible for one molecule, for example, but then those all work together in wonderfully complicated ways inside of your body to make you who you are.
Some traits, like height or intelligence, are the product of hundreds or even thousands of genes interacting. But in other cases, changing just one of those 6 billion letters can have a profound effect.
That can, in some cases, be devastating. And all of a sudden you are very, very sick. And so genetic disorders that people are born with are typically the result of these mutations.
These are things like haemophilia, where someone's blood doesn't clot.
If you get cut, you just bleed and bleed and bleed until potentially you die.
Or Huntington's disease, where the brain stops working properly to devastating effect. Each of these caused by defects in a single gene.
So if you could find a way to go into someone's body and just rewrite that one letter in their cells, you could heal them.
You can see now why he says it's a big deal. Think of our Word document. If there's a spelling mistake in a gene editing is like going in and fixing that. The technique has been around since the 70s, but it's been difficult, slow, and expensive. Then, recently, scientists discovered something called CRISPR.
Clustered, regularly interspaced, short palindromic repeats.
Don't worry, you don't need to remember that. What's important is that it's a process that bacteria use to defend themselves against viruses. They effectively memorize bits of the virus's DNA, Then, when the virus next attacks, the bacteria can edit or chop out the offending DNA and kill the virus. In 2012, two scientists
realized that these bacteria were able to recognize just about any sequence of DNA. And it occurred to them that it didn't have to be the DNA of viruses. It could be the DNA of human genes or tomato genes or whatever genes you wanted. And they found that they could tailor CRISPR molecules to cut any piece of DNA that they wanted.
Remember the DNA Word document? Well, rather than blindly searching through 6 billion letters, we've now got a powerful search and replace function. Scientists can chop out a gene or part of a DNA sequence and replace it with other DNA. It's not perfect, but compared to what they had before,
it's drastically less expensive. So it's just dramatically accelerated things. And it's allowed scientists to try out things they wouldn't otherwise because it is so cheap and quick.
And so far, CRISPR appears to work with any living material. Humans, animals, plants.
CRISPR has really changed everything.
Our first expert witness has explained how the discovery of CRISPR revolutionised the field of gene editing, bringing what was once science fiction much closer to being science fact. But for some, it's driving nightmares, literally. One of the women who discovered CRISPR
was thrilled when she realized that she had found a new way to manipulate DNA. But after a while, that thrill turned to anxiety, and actually she started having bad dreams at night because she was worried that she had unleashed a power that humanity might not be able to control and that might lead to terrible things.
Part 2. The cutting and pasting edge.
In our own institute, we have people making so-called mini brains.
Our second expert witness is Robin Lovell-Badge, a scientist working at the Francis Crick Institute, a biomedical research centre in London. When you say in our lab we have people making mini brains, that's just completely blown my mind in terms of mad scientist kind of possibilities. Just give us a bit of a picture what that really means.
Basically, if you culture these pluripotent stem cells in a particular way, they will organise themselves to form these little organs.
So let's just lay that out in layman's terms. The mini brains are stem cells growing into the earliest phases of brain development.
They're not really brains. They are parts of nervous tissue that looks and organises itself into structures that are reminiscent of what would normally go on during embryonic development.
Scientists use CRISPR to understand the role of particular genes in this process. The Crick Institute is conducting experiments on everything from chickens to yeast to mice, and they're the first lab in the UK to use CRISPR on early human embryos.
To try and get some basic understanding of fundamental mechanisms going on in early human embryos. Of course, the other reason for using these techniques is the potential to avoid serious genetic disease in children. And to do that, you could imagine using the methods to correct a mutation in an early embryo.
In CRISPR research, this is the cutting edge. And also the most controversial stuff.
I suspect we'll see even more labs starting to do it in the UK and elsewhere.
Regulation of research on human embryos varies widely. In the UK, it's allowed but highly regulated. In Germany, it's banned. China has guidelines, but no laws.
Other countries have no regulations at all, and that would include some Eastern European countries, like the Ukraine, seems to have very little in the way of regulations. United States is a big problem because if you have private money, in some states, you can do pretty much anything. It's really quite free what you can do in the US.
As well as embryos, trials are also being carried out using CRISPR techniques on adults, where they could improve treatments for a variety of diseases, including cancer. These techniques are still in their earliest stages, So should we even be concerned about what they can do?
What worries me most is the idea that you'll have rogue clinics operating outside of strict regulations who will offer services which are not justified, not well tested to guarantee their safety, preying on patients or families of patients who have horrible diseases and are looking for any sort of cure.
So for Robin Lovell-Badge, the risks with gene editing are more about peddling false promises than the actual effects. But what about when the science gets better? Should we worry that embryo editing might lead to so-called designer babies?
The possibility of any sort of enhancement or designer babies for trivial reasons is just unacceptable and should not be done because the risks far outweigh the possible benefits.
So, is gene editing out of control? Our second expert witness says there might be risks from rogue operators and that global regulation is patchy. But he says that mainstream scientists see a clear distinction between avoiding genetic disease, which is okay, and designing babies, which is not. But not everyone is ready to trust the scientists.
Part 3. Unknown unknowns.
I was teaching classes at a California State University in the late 1990s when the announcement came that a mammal, the famous Dolly, the sheep, had been cloned.
This milestone in the history of genetic engineering was also a turning point for our third expert witness, Marcy Darnowski.
In that conversation that took off after Dolly the sheep, there was also a lot of talk about genetically designing human beings. And I was shocked.
Marcy Darnowski now runs the Center for Genetics and Society in Berkeley, California. While some of the conversations in the wake of Dolly the sheep shocked her, the future they were talking about seemed a long way off, until now.
What used to be seen as a science fiction hypothetical really is an urgent social justice issue.
She says it's okay to use gene editing to fight diseases like cancer in adults, where any genetic changes aren't passed on to future generations. But she draws the line at gene editing of sperm, eggs, and embryos, known as germline editing, where the genetic changes could be passed on.
If we were to start to use CRISPR on future generations, it wouldn't be a slippery slope. It would be more like falling off a cliff. First of all, it's not safe now and may never be able to be safe enough to be used because you're making these changes at such an early stage that they're going to interact with an extremely complicated developmental trajectory of a human organism. And then there's another really important point. It's not needed. If you're at risk of transmitting an inherited disease, then you have other options for doing that.
These include embryo screening, where multiple eggs are fertilized, then the embryos are screened for genetic diseases, and only unaffected ones are considered for implantation. For the tiny number of people for whom this wouldn't work, Marcy Darnowski argues that allowing gene editing is simply too great a risk, because in practice, the bright red line between fixing genetic diseases and designer babies is really blurred and grey. Are stronger bones or better eyesight medically justified, or designer tweaks?
It would be so difficult to define that line, and it would be next to impossible to enforce it as a matter of policy. That's not a kind of door you can open just to crack. It's a door that once it's open, it's going to blow wide open.
One reason for this is that decisions around fertility aren't just subject to medical, scientific, or ethical considerations. Inevitably, commercial and social pressures enter the picture too. Imagine the ad for a fertility clinic.
Be sure you get your child the best start in life and that kind of thing. And I think people would feel obligated, even if they were uneasy about it, to do for their child what the more affluent person next door was doing for theirs. And I think you would find yourself in a situation of competition, of marketing pressure, and could easily take us down a road we don't want to be on.
Earlier this year, a report by an international committee convened by the US National Academy of Sciences said that gene editing human embryos to prevent diseases could be ethically okay if it's the only possible option for a couple. That door that Marcy Darnowski talked about was cracked open.
I think it was a terrible mistake.
Our third expert witness has argued that the genetic editing of human embryos is both unnecessary and undesirable. But we know it's already happening. So is gene editing out of control?
In the sense that scientists are making statements and in some cases moving ahead, then I would answer yes. Scientists as a group are often inexperienced in really thinking about social issues, about policy issues, and they're very excited about what they're discovering, as they should be. But I think it's very difficult for them to come to terms with the power of this technology because this technology can really shape our entire society.
So what's it like to be caught between the great power of this technology and the great responsibility that comes with it? Time for our final expert witness.
Part 4. The Creator.
Well, I was incredibly fortunate that my parents took me to see the Galapagos Islands when I was 10 or so. And I was just floored by the incredible variety of species there.
Kevin Esvelt isn't the only scientist whose life was altered by the animals of the Galapagos Islands. In the 19th century, it was the birds that set Charles Darwin on the path to his famous theory of evolution. For Kevin Esvelt, it was the iguanas.
And I decided then that I wanted to learn how such marvelous things were created and understand that process well enough to do the same.
To do the same. What does that mean?
It means that evolution has created wondrous forms, most beautiful, and I think it is our responsibility to understand how that is done and see if we can do it at least as well.
And that's now his job. He runs the Sculpting Evolution group at MIT's famous Media Lab in the United States. Back in 2013, he was thinking of abandoning CRISPR research because it was so popular and he couldn't see what he could add. Then, while walking through a park in Boston one day,
I idly wondered, what if we taught the cell how to edit DNA on its own?
Remember, DNA contains instructions for cells to make things. Kevin Esvelt's breakthrough was realizing you could edit a cell to make its own CRISPR system, rather than scientists pasting it in from outside. It's called gene drive, and to cut a long story short, this guarantees that edited genes will carry on through multiple generations, forever. This moment of inspiration was the beginning of a roller coaster weekend.
The first day was total elation, right?
Part of the excitement was that this seemed to be the final piece in the puzzle for existing projects like engineering mosquitoes to stop spreading malaria.
Here was what seemed the key to alleviating so much suffering.
It was also the realization of that dream he had as a 10-year-old in the Galapagos Islands.
I used to go hang gliding, and I would have said before that the feeling of being one with the sky was perhaps the greatest elation that a human could feel. But when you put together the pieces like that and everything just seems to coalesce into a tapestry perhaps almost as brilliant as evolution and natural selection have woven in the ecosystems around us, that comes pretty close, and it may in fact be better.
But the elation didn't last.
The next day I started wondering, well, wait a minute. In principle, this is something that we can't even really test in the wild. I'm not even sure we can test it in the laboratory. And that's because laboratory organisms can escape into the wild.
If that happened, the gene drive would ensure that any modified gene would eventually spread through the entire wild population of the organism.
If you build a standard CRISPR gene drive system in the lab, you just made a decision that could potentially affect other people. And if you do that work without telling people first what you're going to do, you're denying them a voice in a decision that could directly affect them. That's not true of almost any other kind of research. So that's a moral problem right there.
From life-saving discovery to potentially wreaking havoc in the wild, Kevin Esvelt had to decide what to do next.
Frankly, if it had been up to me alone, this would have been an epic disaster.
Why do you say that?
Because no one person is wise enough to make these decisions. I have made so many incredibly stupid decisions, and I have so many blind spots concerning this technology that it really took people wiser than I to show me how the world really is.
After taking advice, Kevin Esvelt decided to put his idea out there and get feedback about safety before he did any experiments. Later, other researchers independently developed a gene drive. They were on the brink of publishing an article outlining how to do it. But Kevin Esvelt's team managed to intervene at the last minute to get them to rewrite the paper, emphasizing safeguards.
So ultimately, it turned out well. I would say it's even a great example of how the scientific community can, to some extent, self-police. But there was a little too much chance in there for my liking. If that ever happens with a truly dangerous technology, we're pretty hosed.
He means that another powerful technology could be discovered and that scientists might get carried away with experimenting before they realize the risks. It's not CRISPR gene drives themselves that Kevin Esvelt is worried about. It's what his experience has revealed about how science works.
Gene editing is no more out of control than any other technology, but we would be far better off if the system by which we make these discoveries and create these new inventions and methods of changing the world were a little bit more open to input from other people.
So is gene editing out of control? In this inquiry, we've heard how the road ahead holds great promise and great risk. This isn't new, but with CRISPR technology, we're now accelerating down that road much faster than expected. Whether we're out of control or not might depend on whose hands are on the wheel. As we've heard, the crucial decisions which will determine how the technology is used can't be made by scientists alone. That's all for this edition of The Inquiry, and from me, James Fletcher. The program was produced by Neil Rozelle and Sarah Shebier. It was mixed by Rod Farker and edited by Emma Rippon. We always love to hear what you think of the program, so please review us wherever you get your podcast or get in touch via our Twitter account at BBC The Inquiry. Next week, we're asking, is it time to ban the plastic bottle?